speaker of today. He's assistant professor at the Institute of Genomic Medicine, where he leads the development of tools and standards for advancing precision medicine and knowledge of genome alterations in cancers. He is also a very strong advocate for open science resources and initiatives. So he's the director of the International Variant Interpretation for Cancer Consortium and also co-leader of the Variant Representation Group of the Global Alliance for Genomics and Health. He's also active in the ClinGen Somatic Working Group and the American Society of Hematology. He co-developed several precision medicine web tools, including the very popular Gene Interaction Database and the CIVIC database for cancer variants. He's also a lead developer of the GH4GH variant representation specification, which will be the topic today. And this is a computational framework for precisely representing and sharing genomic variants across systems. So, Alex, thank you very much for having you here. And now I will just let you share the screen. Thank you.
All right. Well, thank you for the invitation to present here and for that very kind introduction. Yeah, so I am from the Institute for Genomic Medicine at Nationwide Children's Hospital. This is in Columbus, Ohio. So I'm presenting to you today from the United States, and I'm going to be talking to you about the GA4GH variation representation specification, which is VRS or VERSE for short. So we'll talk today about VERSE. I know that you've had a number of different GA4GH talks on this call, so I won't belabor this too much, but for those of you that are unfamiliar with it, the Global Alliance for Genomics and Health is a data standards organization that's really, it revolves around building genomics and health data standards for genomic data sharing within a human rights framework. And many of the well-known bioinformatics standards exist under the GA4GH, including VCF, BAM and SAM, CRAM, bed files, etc. And the way the GA4GH works is it's got this matrix organization between real-world driver projects, which are like large-scale initiatives, and different technical work streams, and as well as a couple of foundational work streams that kind of that support all of the technical work that we're doing. And these driver projects that support the GA4GH, these are real-world genomic data initiatives, and they provide input on the standards, and they really drive the development of the standards needed to share data in the international genomics community. Now, two of the driver projects that are very actively involved in the genomic knowledge standards workstream, which is where VERSE was developed, are the Variant Interpretation for Cancer Consortium and ClinGen. And these groups are really focused around how we assemble knowledge about the clinical interpretation of variants that we observe in patients. Through the structured assemblage of evidence to draw conclusions, and the data structures that represent those higher level assertions built upon computational frameworks and evidence lines from different resources necessitate the development of these standards. And to help make this a little more concrete, what we're really talking about here is addressing the interpretation bottleneck. So here we have our, variant scientist in the center of the screen, who's got, just a mountain of case reports to report out and identify what are the driver, like the likely genomic drivers influencing this patient's disease. To interpret this, they need to look across a number of different resources that describe variants and evidence about variants in a variety of different formats and on different reference assemblies. And it requires a lot of expertise and knowledge about each individual resource, which is non-standardized. It's a very ad hoc process that is also very time consuming. And this creates a bottleneck in terms of our ability to interpret genomes. So alleviating that bottleneck requires that coordinated development of genomic knowledge standards so we can translate information across this federated ecosystem of variant evidence. And one thing that we need to be concerned about is how variant evidence it's disparate, it's fragmented, it's distributed, And we need ways to consistently represent that evidence to make it easy to pull it together, to harmonize it, and to integrate it, to draw conclusions from it. So our solutions then must be interoperable across data structures. They have to be self-descriptive to provide appropriate reference context, and they have to be globally consistent across this federated data ecosystem. And our challenge continues to grow as we see newer and novel references that describe different populations, that fully describe genomes in new ways that we were unable to do before with our common genome reference assemblies, as well as variants that exist on experimental sequences that are very different from the references or the reference sequences that they are emulating. And the idea here is that at the end of integrating all of this evidence, that we're able to come up with genomic knowledge. And this is very complex and it's varied. And it's these types of statements like Huntington's disease appears fully penetrant among patients that have a polymorphic trinucleotide CAG repeat count greater than or equal to 40, right? This is from the ACMG guidelines. That MAVE experimental evidence scores as loss of function, supporting pathogenicity of a mapped variant for Brugada syndrome, or that certain types of fusions with subsequent secondary point mutations that cause a change in the protein sequence can confer imatinib resistance in CMLs. So these are all different flavors of genomic knowledge, and we're looking for kind of a universal framework that allows us to collect all the evidence, assemble these knowledge statements, and be able to share it reliably. To do that, the Genomic Knowledge Standards workstream really works in three separate areas. There's variant representation, variant annotation, and sequence annotation. And today we're primarily talking about variant representation. And specifically, we're talking about verse. So this is like the verse landing page at verse.ga4gh.org, where you can go and you can learn about all the different technical components. The screenshot's a bit dated. This is the version 1.3 landing page. In January, we released version 2.0, which includes a lot of new enhancements that enable support for structural variation and for more verbosity in describing variants and the different references and such that they're derived from. And what Verse allows us to do is provide a generic framework that can help us expand the scope of where we're drawing evidence from, what a variant is, and how we represent it. And what I mean by that is probably illustrated best here, where we think about what kind of goes into a typical VCF file, right? You're starting by genotyping. You're looking for these small differences from a reference assembly. You typically do some sort of high throughput short read sequencing experiment to get those variant calls through some SNV or Dellin's calling algorithm, like Dragon or GATK or something like that. And then these algorithms output their calls for the variants into a VCF file or variant call format file. And this is something that's well known. It's broadly used. I'm certain GHGA has, its own collection of VCF files representing the data from the consortium. And but that represents just a very small sliver of all the different types of clinically relevant biomarkers that we assay for using different types of genomics technologies, which include not just sequencing, but also microarrays and PCR and histochemistry. And then the outputs from those can be scored in a myriad different ways. And for us to be able to reconcile all this, we need versatile, flexible structures that allow us to represent data that come from all these different underlying assumptions about the biomarkers and the assays, et cetera. And this is, of course, in addition to the many resources that exist that provide registered identifiers for variants that represent those variants using similar but distinct data models. The idea here is to provide a consistent exchange format so that the way that variants are represented in each of these can be consistently exchanged and reused and integrated in downstream pipelines. So Verse enables this first and foremost through a core terminology and information model. And this is one of the keys to what makes Verse useful as a standard in this space. It provides that semantic grounding of each conceptual entity that is standalone. There's no implied linkage between components. It's all very explicit, and each component in isolation refers only to the content stored within it. And if that seems a little abstract, we'll get into some specifics in a following slide. But for now, just take away the key message that we have this core model, and around that we're able to put on top of it standardized conventions, a standardized data serialization format using JSON, and then implement these using a reference implementation. And the way that we assembled Verse makes it interoperable then across a variety of different formats. JSON, of course, is mentioned, but Google Protocol buffers is another, or older, more mature technologies like XML. The Verse objects are all very self-descriptive. They contain within them all the necessary information to identify a variant and the reference on which it exists in an unambiguous way. And this allows for global consistency and decentralization. So as a result of this work, we're able to get shared computed identifiers for variants that can work across resources without any prior coordination. So as I mentioned, you can find more about the details of this at verse.ga4gh.org. And there, there's a reference, a citation here where I'm drawing some of the figures that are in this slide deck. You can read more about them at that paper. But I'm going to just go over a few of the key conventions that we use to drive this interoperability and this precision. And the first of these is interresidue coordinates. So this is something that's used by other formats, such as the browser extensible data, or BED format. It's used by, say, the UCSC Genome browser and BED tools for describing kind of genomic ranges and the things that exist on them. And the nice thing about interresidue coordinates is that they actually describe the spaces in between nucleotides. So if these upper numbers here represent kind of T, C, A, and G for 1, 2, 3, and 4, the lower interresidue coordinates talk about the spaces in between, 0 to the left of the first nucleotide, 1 to the right of it. And what's nice about that is that you can get away from some ambiguities that exist in a residue system. And this example illustrates this. So if we have an insertion, for example, using a residue-based system, common conventions specify that you say, okay, the insertion occurs between this A and this G, that's 6 to 7, and the insertion occurs there. And so in that case, you're still preserving the A and the G, but something goes in between that when you're saying from 6 to 7. So the coordinates are exclusive of these nucleotides. The insertion occurs between them and not overwriting them. But then if you're talking about deletion or substitution, again, it's 6 to 7, and in this case, the coordinates are inclusive. So there's this implication that depending on the type of variant that you're describing, the coordinates mean different things. And that can be confusing when you start looking across a variety of different variants and a variety of different contexts. It can create a lot of complexity. Moving to an interresidue system makes it easy because we can just say that the insertion occurs at interresidue position 6, which is, of course, in between these. And then if we're talking about overwriting these nucleotides, like with a substitution, or if we're talking about deleting them altogether, we can simply say 5 to 7, and then that it includes those. So this interresidue convention is a key part of what makes Verse distinct from similar variant formats that are maintained by the GA4GH, like VCF or other community standards like HGVS. And speaking of that, another key convention that differentiates this is the notion of how we address insertions and deletions that are ambiguous in their location. And to illustrate this, you consider a repeating sequence in your reference where you have this CAG trinucleotide repeat, CAG, 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 etc. And then if you were to insert a new GCA into that sequence, where does that happen? And since we can only measure the end state, all we really know is that it got three nucleotides longer. We don't know where those new three nucleotides came from because they could be the first ones, the last ones in the middle, et cetera. And they all biologically mean the same thing. Um, so to address this, different standards have different conventions. If you use VCF, it says, okay, we're going to go all the way to the left of the sequence, and that's where the insertion occurs. So the CAG is at the front. If you use HGVS, you go all the way to the end and you say, okay, an AGC is inserted here. Now, note that's not the same as CAG, and that's because this repeating sequence is not, divisible by three. It has, an additional C at the end here. But again, it's ambiguous as to where that insertion occurs. So the actual sequence itself changes as well. And then in things like verse and also the NCBI speedy format, we basically take the entire region of ambiguity and we say this region has been replaced with a string of, CAG, 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 C. So these conventions allow us to then be exactly as precise as our technology and context allow us to be with regard to the insertion event, and has some nice advantages with respect to searching on ranges where you're not going to miss it by searching here, or if you're using, HTVS by searching here. Instead, wherever you search within that interval, you're going to land on this indel event. Okay. So using those conventions and the JSON serialization, we're able to then convert value objects into identifiers. So we can, take that representation, we can digest it, and we can put it into this coded namespace, where we basically say this is a GA4GH object, it's a variant allele type, and it has this computed digest. And what's nice about that is then when you have different descriptions of what are the same variant on the same underlying reference sequence that might have different names, you still end up with the same computed identifier. It's all driven by the data and not by the names given to the data. And what that means, of course, is then you don't need this prior communication on what you even call your sequences. You just need to have the notion of variants on sequences. And if you have the same variant on the same sequence, you get the same computed identifier, which means you can ask a network of nodes that all agree to just representing their data and computing the digest in this way. Have you seen this variant before? Just by sending along this computed ID, which is it's a powerful way of speeding up queries and indexing variants that you have seen. Now, of course, I'm here telling you about a new variant standard, but you're already familiar with other variant standards, so why should you care? Why are we building yet another variant representation format? And there are reasons, of course, you'd want to use different standards. So in VCF, this was really designed for cohort studies where you have constraints to one reference. You're only looking at the genome, and you want to store all of that in a compressed flat file format. VCF is good for these things. And HGVS, in contrast, is all about conventions for human readability. You can stick these inside, English language paragraphs and manuscripts and clinical reports. And it allows you to represent DNA, but also its consequences at the RNA and protein levels. And verse is really built for a different purpose, which is, you really want to create a common language for variants that works across a bunch of online genomic evidence resources, where that information can appear in complex data contexts, like those genomic knowledge statements we described. And you want to represent your variants across diverse sequences. And so I'll give you some examples about where we've applied verse and what this looks like in the real world. So one of our kind of like large scale implementations of this was with the GNOMAD resource, the Genome Aggregation Database. This database is it's a collection of hundreds of thousands of exomes and genomes that describe variants in basically the general population. And I should say, and I do mean the general population. It's not disease free. It's also not disease enriched. So it gives us the notion of how frequent in just the general population are certain variants. And it's a very useful resource for doing genome interpretation, because if you have a rare disease, if you have a disease that you don't see very frequently in the population at large, then there's probably like if you see a variant in that patient and that variant is very common in the population, it's unlikely to be at a minimum the sole driver for the disease context that you're evaluating. And so what we did with Nomad is we said, okay, how can we represent that evidence, that notion of the frequency of a variant in a cohort in an interoperable way that can work with other similar resources like the All of Us database, and be able to exchange that interoperably. So we started by building a cohort allele frequency model under a different GA4-GH specification called the variant annotation specification. It says here it's a draft standard. These slides are a little dated. This was actually released in June. So this is now an approved standard of the GA4-GH and has this cohort allele frequency model in it. And what this allows us to do is to represent that frequency and the number of homozygotes and heterozygotes and things like that that exist in it and link that to a verse allele. And then that can be used in these more complex evidence structures to drive interpretations, such as this variant is benign for Brugada syndrome, primarily because it's so frequent in the general population. The model looks like this. It's basically a way of kind of packaging all this information together. And the idea here is that there is this verse allele that is the focus of this statement, which allows you to then link this evidence to downstream knowledge statements and allows you to incorporate it in those workflows. Okay. So one of the key findings from this is really, this is a test. Can we really apply this at scale, right? So, there is computation involved coming up with those identifiers, coming up with the serialization format, renormalizing the variants so that they don't use the left shift, but instead that full justification format. All of that takes, some compute. And actually, we found that we were able to do this quite rapidly and at scale in Nomad. And what we did is we used the verse Python library, which is the GA4GH reference implementation, to recompute and store in the Nomad version 4 database 1.89 billion alleles and the verse objects that those alleles represent. Another key area for us has been using Verse to enable data sets that currently couldn't be represented using things like VCF or HGVS. One of these was the MAVE-DB data set. So MAVE variants, these are multiplex assays of variant effect. These are high throughput permutations of different sequences that represent kind of known genes in human organisms or human reference organisms that might drive phenotype of interest. And so, you'll take one of these genes and you'll permute kind of every nucleotide across it to try and get a comprehensive representation of what different variants along the context of that gene look like. But the challenge is that these experimental assays don't have sequences that are like one-to-one with the human references that they describe. There are differences in the sequence representation and identity. And for us to be able to then represent that information, we need like a computable format that allows us to get that native reference context and then do this mapping. So that's what we did. We used Verse to create that reference context to represent the variants on these MAVE sequences, mapped them across, and demonstrated what those look like then on human reference sequence annotations over on the right. And then all of that enabled us to then integrate that knowledge about those variants in these MAVE assays into downstream human genomics resources. Now, this work was recently published. It was described in Genome Biology, but I'll give you some of the key places where this data exists. So first, of course, on MAVE-DB, this is like the primary repository for multiplex assay variant effect experiments. You can find these mapped variants, on any one of the experimental pages by clicking on this Map Variants button. And then that's also been applied to downstream tools. So here are two of them. One of them on the left is the genome to phenotype portal. and this is hosted by the Broad Institute. And here we can see that the MAVE data from this study have been ingested there and allow us to visualize kind of those MAVE findings kind of in a three-dimensional protein structure. So you can see where the variants have the greatest impact on phenotypic observation for this gene. This is TP53. Um, and then similarly, over here in the UCSC Genome Browser, we see those same data, and you can see what those look like on the UCSC Genome Browser tracks. We can see the specific changes for each one of these amino acids at these positions. And then these in blue, these kind of represent the ones where there's a significantly different score, which corresponds to these key domains where the folding occurs for the TP53. Okay. Another common integrate or another well-known integration is the variant effect predictor, or VEP, from Ensemble. Here you can see the MAVE data inside these for any given position that's being queried. You can see the different MAVE score sets for which there is data, and then the corresponding scores. So you can use these to help you interpret this evidence in your variant effect predictions. And then we also have this data integrated into the Decipher interface. So this is a kind of a clinical genomics resource that's used for interpreting variants, uh, and also from Omble EBI. And this the MaveDB section of this resource here provides that additional information from these score sets relevant to the variant being assessed here, a BRCA1 variant. I should also note that we've also, in that same study, integrated this into other clinical genomics platforms, including the ClinGen Link Data Hub and the Shariant platform. Okay, then finally, I'll say that we've also taken Verse and made it accessible for cloud workflows. And one key implementation of this is Verse for the NHGRI Anvil platform in the United States, and the Gregor Consortium, which hosts their data on that platform. This is a rare disease consortium, and so the focus is on these patients that have these rare phenotypes and the variants that drive them. And so our goal was to basically is to use the Verse to enable like integration and collection of evidence on cohort allele frequency from this rare disease cohort to empower genotype and phenotype search tools, and then of course make that accessible to downstream resources like ClinGen and Vic, those driver projects from the GA4GH, as well as other services and workflows implementing these standards. So to do this, we drove some updates in Verse Python, enabling us to kind of back compute Verse IDs for older data sets, increasing support for different HGBS expressions, and simplifying validation and normalization of alleles to make it even faster. We built a Verse annotator, which is it's a cloud workflow designed to simplify the annotation of VCFs with Verse allele IDs. So if you have a VCF and you want to put Verse on top of it, like into the VCF itself, this tool allows you to do that on the cloud. There's a test workspace for any of you that happen to be in the US or are familiar with the Anvil platform. But importantly, this allowed us to then integrate Verse into this cloud-based processing pipeline for the Gregor Consortium. And then these were all added to the joint call set VCFs, making them accessible to Verse-enabled tooling. So if one wanted to replicate this, there are some useful standalone tools that are just openly accessible and can be launched on any cloud environment, like Versix, which allows us to create these indices that use these Verse annotated VCF files to enable like rapid cohort allele frequency calculations on those VCFs. And it's usable in Anvil, but again, this is all open source and could easily be ported over to other cloud environments. So if you have an interest in that, of course, I'd encourage you to go to github.com/gks-anvil to see all of the different tools that we've built for use in cloud environments. All right, so these are just a handful of our current and ongoing implementations of Verse to enable this kind of federated genomic exchange system. I'll just acknowledge briefly the different people that contributed to the development of these specifications and their applications. There are quite a few of them. I'm not going to name them all individually, but I would like to call out my verse co-leads, past and present, including Larry Babb, Andreas Prilich, and Reese Hart, as well as our cat verse leads, which we haven't talked about, which is Daniel Puthawala and Brendan Reardon. Okay, and with that, I think